Hello everyone, so today we're going to be talking about meiosis and some of the ways that it can go wrong. Now, I've talked about this before, but the human body is really good at making sure that things happen the way that they're supposed to. Meaning, if it's DNA during replication, it makes sure that there are no mistakes, and if there are mistakes, sometimes it can be fixed. When we're talking about mitosis and meiosis, making sure those chromosomes get to the right side of the cell. But sometimes mistakes do happen. And that's what we're going to talk about today. Now, mitosis and meiosis are both complex processes. It's, as you know, the first step is DNA replication. So during interphase, DNA is replicated regardless of if we're talking about mitosis up here or meiosis down here. And so in that stage, mistakes can happen. We're going to be focusing on the mistakes that can happen in a later stage, but they can happen there too. And then what needs to happen is the chromosomes are going to line up in the center of the cell. So here you see metaphase, because when they're in the middle, it's metaphase. Then they need to pull apart, and then we're going to end up with two daughter cells that are identical to each other. Now, this phase right here, anaphase, is primarily the phase where mistakes are going to happen during mitosis. If we take a look down here at meiosis, then the same thing is true. The difference, of course, is that meiosis sort of happens twice. So we have meiosis 1 right, let's see, here-ish. And we have meiosis 2. Oh wait, no, I started that a bit early. Meiosis 2 here-ish. And so in meiosis 1, we're going to get PMAT, so prophase, metaphase, anaphase, telophase. And during meiosis 2, we're going to get that all over again. So we're going to get prophase, metaphase, anaphase, telophase. And like I said, our anaphase, or our apart phase, is going to be where those chromosomes are most likely going to be making that mistake that we're going to be talking about. We are also going to be talking about possible mistakes that happen here. And so they can happen here, but recall that, of course, we have two anaphases in meiosis. Let me just find the other one. This drawing looks a little bit different than some you may have seen, and that's okay. And so here we have our two chromosomes being pulled apart. Now, of course, this is a simplified drawing. We're starting out with two chromosomes, and then we have two homologous pairs, and we end up with 4 cells after meiosis. And each of these 4 cells has 1 chromosome. But that's sort of a quick little overview, I guess. Let's move on to the next slide. Now, when we're talking about mistakes during mitosis or meiosis, there are sort of 2 types of mistakes. The first type is called non-disjunction, and I'll get into what that means. And the other type is a mutation. So let me explain non-disjunction first. Now, during mitosis and meiosis, chromosomes need to separate. And they separate during anaphase, because remember, A stands for apart. And when they separate, it's called disjunction, because they're pulling apart. They're creating a junction. That's how I remember it. Now, when chromosomes don't pull apart properly, it's called non-disjunction. And so what you see here is exactly that. What we have is a cell And it looks like our chromosomes, it looks like this is metaphase because our chromosomes are in the middle, which means our next phase is going to be anaphase, and those chromosomes have been pulled apart. Now, what you would ordinarily expect to see is these two blue chromosomes ending up over here. But there's a mistake. And what we actually end up having is only one chromosome over here and 3 over here. So there has been a mistake. There's been non-disjunction. And then we've skipped a few stages, but here's metaphase again. So that's metaphase 2, which would make this anaphase 2. So then these two chromosomes are pulled apart. Now, if let's just pretend, no, actually, I'll keep going for a minute. So then these two chromosomes are pulled apart, and we have 1 and 1, and our end results are 1 and 1. Over here, we have an extra homologous pair, and we end up with 3 and 3, and our final results are 3 and 3. So this is a problem because we should have another chromosome in here, which would mean another chromosome in here, which would mean a sister chromatid here, and another sister chromatid here. So what the numbers that we should have over here are 2, 2, 2, and 2. But the numbers that we actually have are 3, 3, 1, and 1. Now, that's a problem because if we have extra or missing chromosomes, it's going to cause issues with the cell. Because as you know, those chromosomes contain all of the information required for life. So if we're making, and usually that's expressed in proteins, right? And if we're making too much of a certain protein, then that's going to cause certain things to happen. And if we're making too little of a protein, then that's going to cause certain things to happen also. The other issue that can happen during meiosis is a mutation. Now, I'm sure you've heard of a mutation before, and a mutation is basically a change in DNA. Now, there's lots of different ways that mutations can happen. This one is as a result of crossing over. So let's just go back to our last slide. I'll mosey on up. Now, what you can see is right here, let me get rid of some of this. Right here, where this blue arrow is, we have had crossing over, actually here and here. So we have crossing over happening. And basically what that means is one chromosome goes, hey, I kind of like your DNA, can I have it? And then the other chromosome goes, yeah, sure, we can do a swapsies, and that's what they do. And so what you see, if I zoom in, it's a wee bit blurry, is we have one chromatid that's blue, and one chromatid that's mostly blue and a little bit red. And on the opposite side, we have one chromatid that's red, and one chromatid that's mostly red and a little bit blue. So you see that these chromosomes have actually swapped genetic information, making entirely brand new chromosomes. So this crossing over is really important in creating genetic diversity. And usually it's pretty good at happening without errors. Like I said, the body's good at making stuff happen, but sometimes mistakes will happen. And what can happen is parts of it won't cross over quite properly, and so you're missing part of the chromosome. Or maybe you have a little bit extra genetic information. So these mistakes, mutation and non-disjunction. Now, though the body is usually very good at fixing these mistakes or catching them before they get too far, it does happen, and you most likely have cells in your body that have these mistakes. And they can be any cell. They can be your skin, your esophagus, your liver, wherever the cell is. The consequences of these mistakes in mitosis and meiosis depend on when and where in development. The earlier the development, sort of the bigger the problem. The cell can just die out. So again, if you have too much protein because you have an extra chromosome, or too little protein because you're missing a chromosome, then the cell's not necessarily going to function properly, and it might die out. We could also be changed in some way, again, as a result of too much protein or too little protein, and it depends on what protein is being made and where the chromosome is. And we can also appear to be, I'd prefer to use the word atypical maybe. And this might express itself as appearing to be a little bit atypical. And there's lots of different ways this can be expressed in terms of like what you actually see in an individual. These errors can and will cause what we know as just genetic disorders. So when we're talking about these errors, whether it's mutation or non-disjunction, there's a way that we can see them, and that is through karyotypes. So that's the title of the slide up here, karyotypes. And down here is a karyotype. Now pause the video and just observe some things about the karyotype. What do you notice? Okay, so I hope that you paused it. Did you pause it? Okay, let's go with you did. You might have noticed lots of different things about this image. For instance, you might have noticed that they're always paired up together. And if you did, that's a great thing to notice. You may have noticed that they're usually sort of the same height. You may have noticed that they're black and white, and they're sort of like banded. You might have noticed these spaces. I don't know why those spaces exist, so that's not a relevant thing to notice, but you noticed it. So anyhow, this is a karyotype, and basically on this karyotype we have chromosomes. Here's another thing you may have noticed. We have 22, they're numbered 22, and we have an X and a Y. Now the X and the Y chromosome are sort of special because they're known as the sex chromosomes. A female will have XX, and a male will have XY. So here we have a female chromosome because there's two X's, and a male chromosome because there's an X and a Y. Now, usually in a cell, chromosomes are a jumbled mess called chromatin in the nucleus. But they can condense, and we can take pictures of them during metaphase in mitosis. And so after taking pictures of them, we can sort of organize them, and it can tell us a lot of information. So we have to do a few things to the chromosomes to make sure that they do what we need them to do, basically. So we are going to stain the cells, which thereby stains the chromosomes. We have to take pictures of them because usually during mitosis they're not, orderly arranged over here. And then we basically have to cut and paste them into these pairs. So someone has done that for us. And they're organized a few ways. So our largest or our longest is over here, and our smallest is over here with our sex chromosomes at the end. And they're arranged together because these are our homologous pairs. So one of these chromosomes would have come from mom, and one of these chromosomes would have come from dad, basically. And by lining them all up together, we can see a few different things. For one thing, we could see if we had an extra chromosome, let's say we had an extra 15th chromosome, we have an extra one. We would also be able to see if we were missing a chromosome, so let's say we didn't have that chromosome 20, then we could see if there's any extra or missing chromosomes. Hypothetically, we could also see if one of our chromosomes have a little bit extra bit of information, hypothetically. And so we can see any abnormalities in the structure and number of the chromosomes here. With that information, we can predict what sort of disorders an individual might have. So let's take a look over here. Now, we have mitosis pictured here. And so let's say we start out with these 4 chromatids. Then what if we're saying is normal is that we end up with 2 of these, then that's what sort of the goal is, in a way. Now, of course, this is a simplified version, because if we're talking about human cells, then we're going to have a total of 46 chromosomes, and we want an end goal of 46. So it's simplified, but it's really hard to draw 46 chromosomes in a tiny little box, so we usually do simplify it. Now let's take a look over here. We start out with the same number. So let's say if we were drawing a real cell, we had 46. But, okay, we're missing one. There's missing one. There should be another one there, and it's not. And over here we have an extra. So if our goal is 46, then, maybe we have 45 over here because we're missing one. So it's -1. And over here, if our goal is 46, then we actually have 47, so we gained 1. Now, this is a problem because our goal is 46. And there's a bit of terminology with this. We call this monosomy, because we have one chromosome, or one set of chromosomes. And we call this one over here trisomy. I'm going to run out of room. Because we have 3 sets of chromosomes. So if you have an extra chromosome, then you're going to be trisomy. If you're missing a chromosome, then it's going to be monosomy. And just keep in mind, when we're talking about human cells, we started with 46 and the end goal is 46. When we are talking about other organisms, so maybe a mosquito or a dog, they don't necessarily have the same number of chromosomes as us. So depending on what we're talking about, the goal of 46 is going to change. For example, mosquitoes have 6 chromosomes. Now, again, depending on when these errors are happening, the mutation or non-disjunction, it's going to change what effect it has on the organism. When it's mitosis, cells are probably just going to die out, and that's okay. When we're talking about meiosis, it becomes a bit of a bigger problem, because meiosis results in gametes, which, as you know, would be the sperm and eggs cells. And these sperm and egg cells are going to go on to produce a zygote, or eventually a baby. And so together, these cells that have undergone non-disjunction are going to continue replicating. And so any future cells are also going to have chromosomal abnormalities. And so down here, we have meiosis again. We have meiosis 1 here-ish, and we have meiosis 2 here-ish. And you can see here that this is a normal separation, and this is a non-disjunction separation. And you can tell visibly just by looking at it, because here we have all our happy chromosomes that were separated, and here they did not separate properly, because what we should see is maybe one of these chromosomes over here. But it didn't work properly, so it didn't separate properly. Then, once it goes again, so once it divides again, so this one will divide into this and this, and that's a problem, because we have a cell that is missing a chromosome that it should have. So that's non-disjunction, and we should have that chromosome in there, but it's not. Over here, we have a normal separation because we have 23 and 23. Now, our sperm and egg cells should have 23. So earlier we were talking about 46. Well, our goal changed a little bit because our sperm and egg should have 23. And so over here we have 24. Now, is that monosomy or trisomy? Can you remember? Same thing over here. 22, is that monosomy or trisomy? Well, because we're missing one, it's probably going to be mono. And over here, because we've gained one, it's going to be tri. So depending on whether the error happens in anaphase 1 or anaphase 2, you're going to see different results. And of course, here we're talking specifically about non-disjunction because that's when the splitting up of chromosomes doesn't work properly. And non-disjunction of chromosomes is going to cause genetic disorders when it happens in meiosis. And basically, non-disjunction is when you have an extra chromosome or you're missing a chromosome. And that all happens as a result of them not pulling apart properly during anaphase 1 or anaphase 2. And so we end up with an extra chromosome, so we have 24 instead of 23, or we're missing a chromosome, so we have 22 instead of 23. Now, earlier we were talking about how there are two issues that can happen, non-disjunction and mutations. So we've talked about non-disjunctions, and I'm not going to go into the types of mutations in too much detail, but feel free to pause the video and take a look at the slide. But basically, there are different types of mutations, and sometimes it results in adding chromosomes, sometimes it results in, not chromosomes, adding a part of a chromosome, sometimes it results in deleting a part of a chromosome. There's lots of different, sometimes a rearrangement of the chromosome, so it's inverted in some way. There's lots of different things that can happen. What I really want to touch on is that although I say it's an error, it's not always an error. Mutations are often bad for the organism. They can certainly result in cell death, it can certainly result in huge issues for the organism, but sometimes they're positive. Sometimes mutations are actually beneficial in some way or another for an organism. Maybe it causes the animal or organism, shouldn't suggest it's necessarily an animal, to better withstand its environment. Maybe it's better able to retain water. Maybe it's better able to reproduce. Maybe it's faster. Whatever it may be, the mutation can also be positive. And positive mutations are going to drive the process of evolution, quite frankly. So mutations can be good because it gives them an advantage in the natural environment that makes them maybe a little bit more suited to their environment. And over time, if they're more suited to their environment, they're going to be able to survive and they're going to be able to pass on their genetic mutation, which for them in that moment was actually positive. So although these errors can be negative, they can also be positive. So that is it for our lesson. We talked about errors in mitosis and meiosis. We talked about how mutations can happen during crossing over in meiosis. We talked about how non-disjunction is an issue with the chromosomes being pulled apart incorrectly during anaphase 1 or anaphase 2. We talked about how these errors don't always have a huge impact, but sometimes they do. So it kind of just depends. We talked about karyotypes and how we can use those, we can actually see the chromosomes to see what's going on and see if there's going to be any disorders. And I think that's it for today's lesson. So please let me know, as always, if you have any questions, and good luck.